Good morning. So today we are discussing about MHC complex or major histocompatibility complex. Heard about major histocompatibility complex before? Major histocompatibility complex is a protein. It's a surface protein that is developed inside a cell and it will move outside the surface of a cell. Okay. So mostly in major histocompatibility complex is a set of genes that code for cell surface protein essential for acquired immune system to recognize foreign molecules in vertebrates, which in turn determines the histocompatibility. So the key role of MHC molecule is to bind to antigens originated from pathogens and display them on the cell surface for identification of suitable T cells. In short, what is the real function of MHC? MHC will bind with antigen and it will present this antigen to T cells. That is the functions of MHC complexes. So, MHC complexes are developed inside our cells and it will combine with the antigens, that is come inside to the cells, and it will combine with the antigen and it will move outside the surface of cells and it will present these antigens to T cells. That is the main function of MHC complexes. Okay. The human MHC complexes is also referred as HLA complex or human leukocytic antigen. HLA complex, human leukocytic antigens. Okay. Human MHCs are termed as HLA complexes, human leukocytic antigens. And in humans, the MHC proteins are encoded by a cluster of genes located on chromosome 6. As I also already said, MHC complexes are developed inside the cell, and we can see in basic structure of MHCs are originated from the DNA molecules. Okay, there is a chromosome, that is chromosome 6 present in our DNAs, and these chromosome 6 encodes the MHC proteins. Okay, these proteins when it comes to contact with an antigen, it will combine with the antigen and form a MHC complex. These MHC complexes will move to the surface of cells and it will present this antigen to whom? To T cells. That is the main function of MHC complexes. There is mainly 3 types of MHC, that is type 1, type 2 and type 3. 3 types of MHC complexes are there, and we'll study according to the differences. MHC 1 MHC antigens and present to CD8 T cells. It will present antigen to CD8 plus T cells. We have studied already about CD8 T cells. Remember what are CD8 T cells? Cytotoxic T cells. CD8 plus T cells are cytotoxic T cells. Okay, so MHC1 molecules will bind with the antigen and it will present this antigen to whom? CD8 plus T cells. But in case of MHC molecules are visible on the surface of what? Antigen presenting cells. When it combines to the antigen, it will present this antigen to CD4 plus T cells. CD4 plus T cells are helper T cells. What are the functions of helper T cells? What they will do? Helper T cells will inform whom? B cells that there is an invader or an pathogen is present in our body, and helper T cells will inform B cells and B cells will produce plasma cells and plasma cells produce antibodies. But in case of CD8 T cells, what they will do? CD8 T cells or cytotoxic cells will release cytotoxins and it will kill the antigen immediately at the point where cytotoxins are released. But in case of CD4 T cells, these T cells will inform B cells that there is an invader in our body and it will destroy by informing the B cells. B cells will produce plasma cells and from plasma cells it will release antibodies. Understood? Okay. And one more thing, one more difference is there. MHC1 complexes will act with intracellular antigens. MHC2 complexes will react with cellular antigens. What's the differences between intracellular and extracellular antigens? What they will do? Intracellular antigens are the antigens that is developed inside the cells. Inside the cells, like in some bacterial, some viruses will, viral proteins will develop inside the cells, and as I already said, MHC complexes are developed inside the endoplasmic reticulum of a cell. Okay, from there, these antigens, that intracellular antigens will combine with the MHC1 complexes, and that combined MHC molecule will move to the surface of cell. Okay, here also same thing is going to happen. In MHC2 complexes, the extracellular antigen means it is present outside the cell. Then how it will be possible to come inside? Then here what happens is, extracellular antigens will get engulfed by the antigen presenting cells. Understood? Antigen presenting cells will get engulfed, or it will drag that antigen inside the cell, and it will get combined with the MHC complexes, and again same thing, it will move to the surfaces of cells, antigen presenting cells. And next what happens? After that, these antigen and MHC complexes will present this antigen to whom? To whom? T cells. Okay, T cells. In MHC1, it will present to CD8 T cells, and in MHC2, it will present to CD4 T cells. Okay, that is the difference. This is the structure of MHC1 molecules. MHC1 molecules. As you can see, there is a three different parts, that is alpha region, there is and beta region. Okay, in alpha region, that is directly connected to the cell. This is the cell surface. Okay, MHC complexes are visible on the cell surfaces. This is the just representation of cell, and it is connected to alpha cells. Like there is a 3 alpha regions and 1 beta region in MHC1 complexes. Okay, so MHC1 molecules contain 45 kilodaltons of alpha chain associated non-covalently with the 12 kilodaltons of beta microglobulin. These alpha regions are covalently connected, and they are not connected, that beta molecules are not connected. Okay, then the A chain is anchored in the plasma membrane. As you already seen in the picture, the A chain is anchored in the plasma membrane. This is the plasma membrane of the cell. Okay, anchored in the plasma membrane, and it is hydrophobic transmembrane segment and hydrophilic cytoplasmic tail. Okay, there is 2 regions in this plasma membrane. This is hydrophobic region, and this tail is hydrophilic region. Okay, so then comes to the MHC class 2. Before that, you have to see the image of MHC class 2 proteins. Okay, MHC class 2 proteins are arranged like this way. There is 2 beta chains and 2 alpha chains. Both these alpha chains and beta chains are directly linked to the plasma membrane. Okay, so understood the concept? Okay, what is MHC molecules? It's a major histocompatibility molecules. Okay, what is the function of it? What is the like clinical applications of MHC molecules? Where do we use this? Have you heard about transplantation rejection? What is that? How it is applicable in MHC, or how it is connected with MHC? We have heard that during transplantation of an organ, we should match that donor and recipient's organs. Then what is that means? Like what should be matched? These MHC complexes should be matched, because if I am the receiver or I am the recipient, my donor's MHCs should be matching with my MHCs. Otherwise, my MHC molecules will identify those MHCs as antigens and it will produce immune reactions. Understood? So MHC molecules are important. Then